Okay, I think we'll go ahead and get started. Good afternoon, good morning, and good evening to everyone, wherever you may be logging in from around the world. I am Rob Burgess. I head up business development for Sinobiological, and I want to let you know that we have an excellent speaker today for this installment in our series titled BioTalk Tuesdays, where we hear about cutting-edge technology, and research in the pharma and biopharma sector, as well as in academia and in clinical research as well. I will introduce our speaker here in just a minute, but I did want to mention we have one housekeeping issue. We ask everyone, if you have good questions for our speaker, that you simply type them into the chat box, and we'll withhold all questions until the end of the seminar, and then I will walk through them verbally and try to get to as many questions as I can. Also, if you don't mind, just type in the chat box and let us know where you're logging in from. We always like to know from where people are coming and logging in to our webinars from all over the world. So please let us know your location and the institution that you're affiliated with. Now it's my pleasure to introduce our speaker to everyone. His name is Dr. John Erickson. John is the co-director of the Center for Perinatal Immunity, an attending physician in the Division of Neonatology, and an assistant professor at the University of Cincinnati Department of Pediatrics at the Cincinnati Children's Hospital. This is where he leads a basic science laboratory within a division of neurology and the Center for Inflammation and Tolerance. John's research focuses on immunological adaptations during pregnancy that enhance neonatal defense against serious infections and promotes maternal-fetal tolerance. A significant discovery from Dr. Erickson revealed that during pregnancy, maternal antibodies undergo subtle modifications in their N-linked glycans, particularly the deacetylation of terminal sialic acid residues. This modification enables antibodies to protect against intracellular pathogens by binding to the Siglec CD22 on neonatal B cells, thereby reducing the production of the immunosuppressive cytokine IL-10. His lab's ongoing work integrates immunology, glycobiology, and reproductive biology to develop strategies that reduce neonatal mortality and improve child health worldwide. I should mention that John obtained his MD and his PhD from Vanderbilt University. And the title of Dr. Erickson's talk today is Pregnancy Enables Antibody Defense Against Intracellular Infection. John, it's a pleasure and honor to have you, and now we will turn over the screen to you.
Great, thanks, Rob, for that really kind introduction. And appreciate everyone at Sinobiological for the invitation to give this talk today. And also appreciate their cutting-edge research that they're doing there, and production of multiple reagents that our lab uses that are kind of indispensable for the work we do. But I'm happy here today to talk more about our research and then get any questions from the audience. So I'm going to launch right into the talk today. So a little bit about what we're going to cover, just so we kind of know where we're going. I'll just give some background for those of you that aren't as familiar with either maternal or neonatal immunity. I'll give a little background on our particular interest in glycobiology as well. And then we're going to talk just about some of the things we've learned by studying immunity through this lens of pregnancy, and then a new mechanism for antibody-mediated protection that we've uncovered through this work. At the end, we'll have some time to conclude and talk about future directions, and then I'll answer, as Rob said, as many questions as we can from the audience. So first, I want to talk a little bit about something that's near and dear to my heart. So I'm a physician scientist. My clinical training is in neonatology, so we take care of sick babies. Unfortunately, a lot of babies die every year, and this is not something that we hear much about in the news, unlike the COVID pandemic and some of the other more press-grabbing things that happen. But under 5 mortality is still a big problem. About 5 million kids worldwide die every year. Neonates then are these that actually make up about half of all this mortality in kids under 5. So a very tiny age range makes up a lot of the mortality, because neonates are particularly susceptible to a lot of morbidity and mortality. So this comes out to be about over 2 million deaths per year, so which is kind of a staggering amount. And so in thinking about a lot of the causes for these, I've highlighted a few here that are infection related. There's also a lot of birth trauma and things that are a major contributor. And a big piece of this is prematurity. And then this prematurity umbrella also encompasses infections that occur during pregnancy that lead to prematurity, as well as premature infants are at even especially high risk of infection. So what we're talking about today is clinically very relevant and something that we'd like to be able to make a dent in through our research. A little bit of background just about adaptive immunity and kind of where our research fits in. So neonates on their own are born in a relative state of immunodeficiency in that they don't have mature T cell responses or host defense. And so they kind of are critically reliant on the antibodies that they get from moms. And so I'll talk a little bit more about how that antibody transfer occurs. But as we know, antibodies are classically thought to have multiple mechanisms to protect against extracellular infections. So either bacteria or parasites in the extracellular environment or viruses while they're in kind of their extracellular phase of their life cycle. For pathogens that reside inside of cells, classically we think of the cell-mediated immune response as being more critically important for that protection. So CD4 and particularly CD8 T cells that can target these intracellular pathogens. And while there is some research that has uncovered some different mechanisms by which antibodies can protect against intracellular pathogens, this research has been limited, and for some pathogens, it's really thought to not occur at all. And so our research then is focused in on this area of this critical window where antibodies are really required for defense against infections. To go a little bit deeper, I'm not going to go into all the details of this, but there's a well described and evolutionarily conserved system to transfer antibodies across the placenta to neonates. And this is so that neonates are then born with the maternal repertoire of antibodies to protect them. And so this system requires Fc neonatal receptor and Fc gamma receptors to kind of selectively move antibodies over to babies to help protect them. And this doesn't stop postnatally because antibodies continue to be transferred via breast milk. So in humans, this is predominantly IgA, although there's some IgG. In rodent models, it's primarily IgG, but these antibodies in breast milk can then also be transported into the neonatal circulation and help protect against infections. And so this is all very important because there's this kind of, as I mentioned, critical window of susceptibility where babies are born and are at very high risk of infection. And as I showed before, that can lead to a high degree of morbidity and mortality. Once babies, reach a certain age, 2 to 4 months, we're then able to immunize babies using the collective armada of vaccines that we currently have that provide a lot of immunity. But during these first few weeks and months, the antibodies transferred from mom to baby are what's critically important to protect them. And so then that therefore means that whatever repertoire of antibodies moms have then can be transferred to babies. But moms have to first have these antibodies, of course, to be able to protect the baby. So because of that, there's a whole, a field of maternal immunization that's started decades ago, but has continued to build. So it's been known for a long time that you can prevent neonatal pertussis and tetanus by immunizing mom with like a Tdap vaccine during pregnancy. Influenza can prevent, the influenza vaccine can prevent lower respiratory tract infections in babies. And then more recently, the COVID vaccine, and now there's a FDA licensed vaccine for RSV. All of these with the idea of if you immunize mom, you're going to get this kind of halo of protection for baby. Of course, coming down the pipeline, there's a vaccine potentially for Group B strep. But then outside of that, there's still numerous pathogens that cause a lot of clinical disease for which we're still in the preclinical phase, or there's really nothing in the pipeline to help protect babies and pregnant moms against infections. And again, this is particularly important in highlighting the morbidity and mortality associated with intracellular pathogens. This has been known for some time that there's actually enhanced susceptibility of pregnant moms, the fetus, and neonates to pathogens that are really adept at hiding inside of cells. I'm not going to go through all of these, but primarily my purpose here is to focus on the pathogen that we're going to use to study this biology throughout the rest of my talk, which is located here at the center of this because it can cause disease in all three of these populations, which is Listeria monocytogenes. So Listeria has been used for a long time as a model organism, but of course it's a clinically relevant pathogen too. If you've heard in the news recently, there is currently a Listeria outbreak contaminating everything from meats to frozen waffles to vegetables. And so a lot of plants have been shut down and a lot of foods are currently unavailable because of Listeria outbreak. This fluorescent image is showing kind of the classic Listeria biology, where Listeria gets into a cell and then it uses one of its virulence factors called ACT-A to polymerize actin, and that's how it polymerizes actin and propels itself around the interior of a cell. And so because of this, once Listeria gets inside of a cell, it's very good at staying in that cell, and it will then spread directly to an adjacent cell and avoid the extracellular milieu. And so because of this, it's been thought for really decades that antibodies play really no or a very limited role against Listeria. And Listeria has been used as a model to study adaptive immunity through CD8 T cells pretty extensively by several groups. But again, this has not been well studied during this critical window of susceptibility, which is during pregnancy. So one of the questions we had was, does pregnancy modify these classical immunological tenets that we kind of have already discussed? And if so, how does this happen? So that was kind of the impetus for launching this project and some of our initial work. So from there, I'm going to transition now to the background, now that we have that kind of foundation, and talk more about the revelations that we've had from kind of studying this through the lens of pregnancy. So first, let me just say, as I kind of alluded to, a lot of the tenets of adaptive immunity and how it functions has really been established by using adult rodent models, particularly prime challenge models. And so I'm going to give one example of that. This was an experiment we recapitulated in a recent publication. But in this type of experimental setup, we use an attenuated strain of Listeria. So it's lacking this virulence factor called ACT-A that allows the actin polymerization. So because of that, it's highly attenuated. It can't spread cell to cell, but it still primes a very robust adaptive immune response, including antibodies and T cells. So we can use this to prime mice safely. It's rapidly cleared. We generate immune response, and then we can directly challenge these adults with the virulent strain of Listeria that's able to polymerize actin, and then quantify the amount of bacteria 72 hours later. And then we can do this in a variety of immunodeficient mice to characterize the different adaptive immune components. So for example, in MUMT mice that lack B cells, we can assess the role for B cells and antibodies. And CD8 knockout mice, we can assess the role for cytotoxic T lymphocytes. And so what we found really recapitulated what's been seen before in the literature, that when, and this is showing bacterial burdens in the spleen. We also usually look in both the liver, and this is log 10 transform. So this is 10 to the 8th bacteria in a naive wild type mouse that gets infected. However, if that wild type mouse has previously been primed with attenuated Listeria, you can see near sterilizing immunity. And then when you do a same experiment using a B cell deficient mouse, you get the same result. So really in adult mice, this is part of the reason it's thought that antibodies and B cells really play very little role in protecting against this intracellular pathogen. However, you now see a noticeable decrease in the ability to protect against Listeria when these mice lack CD8 T cells. So therefore, this is a lot of the type of experiments that have shown before about the critical role for CD8 T cells. But now what we wanted to do was to kind of shift this paradigm and look at this through the lens of pregnancy. So we used a very similar model. We're now using female mice that are these 3 different genotypes, priming with our attenuated strain of Listeria. And then we get these female mice, a few weeks later, we get these female mice pregnant by a wild type male, so that then the offspring are phenotypically wild type. And then we challenge the neonates with the virulent strain of Listeria and see what the protection looks like. So using this model, we see things are flipped on their head. It's very different. So first of all, shown in blue here is this is just the susceptibility of neonates to naive, that are born to naive dams, versus if the dam has been primed with Listeria, we can now see that there's about a log of protection in the liver, and we saw a similar thing in the spleen. So this shows there is actually vertically transferred immunity against an intracellular pathogen. However, when we look at the different immunodeficient mice, when you challenge offspring born to a B cell deficient mouse, you now lose protection. So this is the opposite of what we saw in adults, where B cells were dispensable. They're completely indispensable in this vertically transferred model. Whereas when you do the same experiment with CD8 deficient moms, there's still protection. Thus, this suggested to us that really in a vertically transferred model, it is the antibodies that are providing this protection and not CD8 T cells. As opposed to, instead of just looking at bacterial burdens, we've also done this looking at survival. And we see that wild type mice born to a naive dam are highly susceptible to this Listeria infection. So you can see about 90% mortality. And when the dam has been primed but lacks B cells, there's no protection against mortality. versus when a wild type dam has been primed, we now see protection. And this to about 75% survival. So we kind of flip this mortality on its head when there's antibodies present and are able to protect neonates. Moving aside from this kind of vertically transferred model and to enable us to do more mechanistic studies, we've also validated that passively transferred antibodies can recapitulate this. So to do this type of experiment, again, we primed mice with our attenuated strain of Listeria, and then we bled these mice to obtain antibodies when they were in the virgin state. And then in another cohort of mice, we got these mice pregnant. And then late in gestation or early in the postpartum period, we bled these mice to obtain the serum from these pregnant mice. And then we either passively transferred the virgin sera or the pregnant sera to neonates, and then we challenged them with virulent Listeria. From this, we found that compared to the transfer of naive sera, when you transfer antibodies from the virgin mouse that's been primed with the attenuated strain, there's no protection. So these antibodies alone are not able to protect. However, when that mouse has gone through pregnancy and we pull these antibodies out and transfer them to neonate, the antibodies are now able to robustly protect the neonate against infection. Thus, all the magic is happening during pregnancy, and it's happening through antibodies. So from this, we decided that we needed to get more deep into the details about what was mechanistically happening to the antibodies during pregnancy. Of course, we started by looking at some of the more obvious things, like is the specificity of the antibodies changing? Is the titer of the antibody changing? The avidity, the neutralization ability, a lot of the classical things you think antibodies do. And really, we quickly crossed a lot of those off the list because there was no difference in the virgin mice or the pregnant mice. We did some other experiments where we transferred the virgin antibodies that normally didn't protect when you just give them straight to a neonate. But if we transfer them to a nursing female and allowed her to give the antibodies to the neonate, we then saw protection was now enabled. So this suggested, as well as our negative data before about the other antibody features, that there was a post-translational modification that was occurring to these antibodies that was likely responsible for their protection. So for those that know antibody biology really well, you know that antibodies are actually glycoproteins. And this is showing the potential IgG N-glycan sites or N-glycan sites on an IgG molecule, which is more simple than some of the other IgG isotypes, but for Ig subtypes. So for IgG, 100% of antibodies are glycosylated on a conserved asparagine in the Fc region, and then about 10% of all comers of antibodies are glycosylated in the FAB region. And while this is a conserved N-glycan site, most of these N-glycan sites in the variable region arise via somatic hypermutation because there's very few germline encoded N-glycan sites. And looking about kind of just the general frequencies of the modifications to N-glycans, most Fc glycans are fucosylated. And then as you kind of move towards more mature structures, you see about 50% to 2/3 get galactosylated to some degree, and then a minority gets sialic acid added as a terminal modification. This Fc glycan has been fairly well characterized for its functional effects on antibodies. And largely that has to do with the ability of the modification of the affinity of the Fc region for Fc gamma receptors. So these kind of less mature forms of glycans have a higher affinity for Fc receptors, and they're better at things like ADCC and cytokine release, versus the ones that are more mature, particularly those that possess terminal sialic acid residues, are thought to be more anti-inflammatory. And again, this is just a schematic showing of kind of where these N-glycans on the Fc are tucked down near the the binding site to the Fc gamma receptors. The function or the role of the FAB glycans is not quite as well described. Just looking at the types of N-glycans, we know they tend to be a little bit less fucosylated and that they're definitely more sialylated than their Fc counterparts. And there's been some research that shows that since these N-glycans are in the variable region, they can either affect antibody affinity, either decreasing or enhancing antigen binding. They can hinder other protein interactions or affect the half-life of the antibody. Outside of that, it's not very clear what the role of these FAB glycans are, but we're going to talk more about some of how our research has expanded upon that. So when we started to look at the N-glycan profile for Listeria, we were honestly expecting a more of this inflammatory subtype that's able to do ADCC and some of these more Fc effector functions. However, what we found using a lectin array and different individual lectins is actually most of the glycan profile for Listeria-specific antibodies look like this, meaning that most of them were sialylated. And also we proved by running these antibodies over an SNA column, SNA is a lectin that binds specifically to sialic acid, that actually the majority of the Listeria-specific antibodies are sialylated. So that was an interesting finding. So we wanted to dig deeper about the role of this sialic acid in the protection against Listeria infection. So in order to do that, we first purified out the IgG fraction from pregnant mice. So we knew this is the IgG fraction that we know is able to protect neonates against infection. From this purified IgG, we then treated it with neuraminidase to remove the sialic acid group. And then we transferred equal amounts of these to different groups of neonates to then see what was protective by challenging them with virulent Listeria. Interestingly, we found as compared to just naive IgG, when we transferred the IgG fraction from pregnant mice, we recapitulate again what we saw before with the serum, that this IgG is protective. However, interestingly, when you remove all of the sialic acid, you completely lose the protection. Thus, the sialylation is critically important for protection against Listeria. Then what we did was a complementary gain-of-function experiment where we then used a glycosyl transferase called ST6-gal1 plus the donor sialic acid species nu5AC, which I'll talk a little bit more about in a second, but one form of sialic acid, to re-sialylate these antibodies that we desialylated. And then we found functionally that these antibodies are now able to protect again once you re-sialylate them. So the question then became like, why then doesn't this IgG from virgin mice that kind of by lectin analysis looked very similar to pregnant mice not protect if it also has sialic acid? So to look at that, we had to start to think about, well, what does sialic acid really mean, and what are the different types of sialic acid? So sialic acid is a more general term that refers to several different species. And so one of those is nu5AC, or acetyl acid, which is the primary form of sialic acid in primates, which includes us. We've lost the gene that converts from nu5AC to nu5GC, so humans primarily possess this form of sialic acid, whereas in mice and most other mammals, there's either equal amounts or for antibodies in particular, actually there's a predominance of nu5GC or glycosyl acid. And really these structures are very similar, as you can see, except for the addition of this hydroxyl group for nu5GC. So from this, what we decided was we wanted to see then, could we interrogate these different types of sialic acid using the virgin antibodies? So we know that the antibodies from prime mice that in the virgin state do have some form of sialic acid, which we showed by lectin array, but really weren't protecting mice. So we then set out to do a similar experiment as what we did with the pregnant antibodies, where we treat with neuraminidase to kind of remove all the sialic acid and start from a clean slate. And then we either added back nu5GC or nu5AC. And then we transferred this to neonates and then challenged them with Listeria. Interestingly, what we found here, again, is that the IgG from virgin primed mice does not protect compared to naive IgG. And then we had another control group where we did not desialylate the antibodies. We just tried to force on more sialic acids. That's what this no neuraminidase group is. And when we did that, also this fraction didn't protect. But when we removed the existing sialic acid and added back either nu5GC or nu5AC, we now saw that we transformed the virgin the virgin antibodies from non-protective to protective. So that was really interesting, suggesting again that there's some type of sialic acid on this virgin antibody, though, that's not protective. So we had to do a little deeper than thinking about, well, what other types of modifications can occur to sialic acid? And so aside from this nu5AC-GC, the next most common modification to sialic acid is 9-acetylation. So in the 9 position, you can get the addition of a small little acetyl group here, which has been shown by other groups to modify the binding of some of the sialic acid to different sialic acid receptors, although other rules for it's not very clear. But we wanted to first interrogate this. So then what we did was, instead of adding back these other sialic acid derivatives, we used the acetylated version of nu5AC to add back. And then we transferred this to neonates. And again, now compared to the non-acetylated forms of sialic acid, when you transfer the acetylated variant, you no longer have protection. So this suggested to us that the acetylation of the sialic acid could be the difference then between what's happening in virgin mice and pregnant mice. We did, though, need to set out to prove this more conclusively. So what we did was then moving away from lectin assays, which are very helpful, but are not, of course, the gold standard for identifying these glycans, as we turned to a mass spec approach. We collaborated with folks at the Complex Carbohydrate Research Center down at University of Georgia, and Parastu Azadi's group, to do this experiment. In our hands, what we did, though, was purified the antibodies. So we took either the immune serum from virgin mice or pregnant mice. We incubated it with Listeria, and then basically eluted off the Listeria-specific fraction of antibodies, ran it over protein G, repurified it a few times to clean it up, and then we had this Listeria-specific prep that we then sent to them to do a mass spec analysis on. The first thing we noticed when we looked at the conserved Fc glycan was that there's actually nothing very exciting going on. A lot of the Fc glycans had the kind of a typical either mono or digalactosylated and very little sialic acid on the Fc region. And this was similar between the virgin and the pregnant preparation of antibodies. However, what we then noticed was that aside from the conserved Fc N-glycan, we noticed several different glycopeptides that were not the conserved N-glycan, so that were presumably coming from the variable region, that did possess acetylated sialic acid. And so what's shown here is 3 examples of these glycopeptides, and then looking at the mass peak for acetylated nu5GC. And what we saw was a much greater peak in the virgin sample compared to the pregnant sample for all 3 of these different glycopeptides. And then we also did experiments where we cleaved off the Fc and glycan to just enrich for the FAB glycans, and we preserved this finding, really highlighting that these were from the FAB region. And so really what that showed us was that the difference between these 2 samples, as far as everything we could tell by mass spec, was that the FAB N-glycans from the virgin mice possessed this acetyl group on the sialic acid shown here, versus there was about 10-fold to 20-fold less, depending on the glycopeptide, in the pregnant state. So we wanted to then show functionally this is really what was responsible for our difference in protection. Oh, sorry, I jumped the gun. Before we went to the functional study, we wanted to confirm using other means that this was indeed the result. So instead of doing mass spec, we actually have a panel of virolectins that have specificity for these sialic acid modifications. We originally obtained this from Colin Parrish's group at Cornell, and then subsequently Sinobio has helped us produce more of this, and we've tagged it in multiple different ways now to help with using this as a probe in multiple different assays. But when using this virolectin, which has high specificity for 9-acetylated sialic acid, we've confirmed that in virgin mice we can detect using this virolectin in an ELISA-based assay this presence of this acetylated sialic acid, versus when we replace it with nu5GC or nu5AC, the signal goes away. But when we replace the sialic acid with the acetylated variant, again, it comes back. And then we can deacetylate it in vitro, and the signal goes away. And then also we have a mutant version of this probe that is mutated at the binding sites. This is a nice control to show specificity of this lectin probe. From here, we wanted to move this into a functional approach in our neonatal model. So what we did was we took these preparations of IgG from either virgin or pregnant mice. And first what we did was use pepsin to cleave off the Fc region to really isolate the biology and the function of just the FABs. And then from here, we further treated this the virgin sample that has this acetyl group on it with an enzyme called SIAE. I'll talk a little more in a second, but this is an enzyme that specifically removes acetylation from sialic acid. And then using our virolectin approach, we kind of confirmed that this enzymatic reaction was successful. And so then we had a preparation of the virgin fabs that had been SIAE treated and deacetylated. So we took all these, purified enough of these preparations to transfer to neonates and then challenged them with Listeria. And what we found was that the FAB region from the pregnant mice, where it had been naturally deacetylated, protects neonates against infection. Just the fabs from virgin mice, again, that have this acetyl moiety, do not protect. But when we deacetylate them in vitro and remove that acetyl group, now these virgin fabs are able to protect. So really, the protection that we were seeing was all the whole business end of this. The molecule was on the FAB region, and it's this FAB N-glycan acetylation that's really controlling everything. So here we wanted to prove that this deacetylation is what was occurring during pregnancy. And so as I mentioned, there's one known sialic acid deacetylase called SIAE. So we confirmed, there's not many tools to study this, so we use just a gene expression approach here, but what we found was that in virgin versus pregnant mice, that we saw an upregulation of SIAE gene expression, and this is in splenocytes. And then also we have collaborated with some folks at UPenn. We have a cohort of non-pregnant and pregnant women during a few different time points during gestation, but we looked at the PBMCs that we had from that cohort and saw that in PBMCs in pregnant humans that SIAE gene expression also went up, paralleling what we saw in the mouse model. We then generated an SIAE knockout mouse using CRISPR-Cas9 technology and with the assistance of our transgenic mouse core. And then using this mouse, what we did was we transferred the nursing females that are either wild type or SIAE deficient, this virgin serum that has the acetyl group, hypothesizing that the wild type mice would still be able to potentially deacetylate it versus this mouse would not be able to. I'm not showing that IgG deacetylation data, but just the functional data, which prove that point, which is when you take these virgin antibodies that are normally, when we directly transfer them to neonate, not protected, but passage them through pregnancy, they get deacetylated. They're now able to protect neonates. However, when the nursing female lacks this key enzyme SIAE and can't deacetylate the antibodies, they're no longer protective. So just to summarize so far, we found that pregnancy revealed to us that antibodies are critically important for vertically transferred immunity to intracellular pathogen like Listeria. Post-translational modification of this Listeria-specific IgG is what's enabling protection. And really, one of the key ways that Listeria has been able to evade antibody-mediated immunity is really because of modification to these FAB glycans. It's not so much that it's hiding inside of a cell and the antibodies can't get to it, but it's more upstream of that when you have this particular acetylation moiety on the antibodies, they're not able to protect. And furthermore, we found that SIAE is the gene or the glycosylation modifying enzyme, in this case, an acetyl esterase that's upregulated during pregnancy, and that mediates this process during pregnancy. So from here, we found kind of this very interesting biology and phenotype, but really we wanted to figure out then how exactly is this kind of subtle change to the antibody and glycan having such a profound impact on the ability of these antibodies to protect. So for those of you that study antibodies, obviously antibodies are diverse molecules that have many different functions that are really remarkable in their ability to protect, including neutralizing toxins and microbes and fixing complement and Fc gamma receptor mediated functions. So all of these have been well described and are super important. And data I'm not going to show you, we started targeting these things right away. We've used Fc gamma receptor knockout mice and complement deficient mice, and we saw that our antibodies were still able to protect. So we had to think beyond that. We really had to think more about what the specific modification of this N-glycan could be doing. So, these N-glycans, a lot of them have very then specific receptors, sialic acid binding receptors, a whole family of them called Siglec, that specifically recognize different forms of sialic acid. And in particular, thinking about the type of sialic acid that's present on antibodies, in humans, that would be this nu5AC that's alpha-2,6 linked to galactose. In mice, it's the nu5GC that's also alpha-2,6 linked. So in thinking about what receptor then kind of specifically can bind to alpha-2,6 linked sialic acid. And one of those key molecules is CD22 or Siglec 2, which is known to bind to these. And actually, this is showing a structure of CD22, but kind of wanted to zoom in on the ligand binding domain. So this is showing, and I'm just kind of orient based on these little cartoons, how I've been showing this before, of how this kind of the sialic acid, which kind of fits down into the binding pocket on CD22. And so you can see how sialic acid kind of nestles right down in here. And in work done by Ajit Varki's group previously and others, they had shown that when you acetylate this 9 position of sialic acid, you actually can completely block the binding of sialic acid to CD22. So it's really a remarkable specificity. So we thought potentially this, although a subtle modification, this could be how it's functioning in vivo. So the first thing we wanted to do was just say, is this the case? So, I mean, CD22 knockout mice had already been generated and readily available. So we used our antibodies from pregnant mice. So again, shown here, these antibodies are able to protect when you directly transfer them into neonates, robustly protect against infection. However, when we transfer these same antibodies to CD22 deficient neonates, we now lose protection. So it did seem that CD22 is absolutely required then for these anti-Listeria antibodies to protect, which fits with the previous data I showed you where sialic acid is also absolutely required. How then is this working through CD22? So I don't have time to talk about, been a lot of literature about CD22, but just briefly about what CD22 normally does is CD22 is expressed primarily by B cells. There's been some exceptions shown in the literature more recently, but CD22 is known to be a negative regulator of B cell receptor signaling. Normally, CD22 binds to either itself or other sialylated glycoproteins and kind of sequesters itself away from the B cell receptor. However, if you kind of fast forward to now an activated BCR where the cell is seeing antigen, CD22 can then migrate in lipid rafts towards the BCR, and it recruits a phosphatase called SHP1, which dephosphorylates key signaling intermediaries in the B cell receptor signaling pathway. And so because of this, when CD22 is ligated, it can decrease BCR signaling and decrease activation of B cells. So the question was, how does B cell inhibition then help protect against Listeria infection? This is a bit counterintuitive, and it comes to a double negative. It must, then what we were thinking of, well, it must inhibit an inhibitor. And so I know that's always a confusing, so we'll talk about, well, what type of inhibitory cell subset then could be expressing CD22? And this is how we became then focused on regulatory B cells. So another big topic that I don't have time to talk all about, but B-regs are, T-regs have been super well described, and there's several groups now that are doing exciting research looking at B-regs and their role. These are diverse cells that are very interesting. They play a role in many different disease processes. But I just want to highlight here that one of the key things that you see that B-regs do is secrete IL-10. So that's what's shown in green. And in many of these disease conditions, IL-10 is important. They also do things like IL-35, and they express PD-L1, other inhibitory ligands, but IL-10 is thought to be kind of a very defining function of a lot of subsets of B-regs. So what we thought was that, going back to our model here of acetylated sialic acid blocking signaling, if you don't have this and you have intact sialic acid, that this could cause BCR inhibition, particularly of B-regs, and given that their key function is to produce IL-10, this might lead to a decrease in IL-10 production. And I didn't say it before, but IL-10 is a key inhibitory cytokine. It suppresses numerous other immune responses. It's really important for counter-regulating pro-inflammatory immune responses, and important for preventing autoimmunity. But in the context of infection, it could be suppressing other immune responses that are helpful for clearing the infection. So we wanted to do a set out to explore in vivo whether or not this was the case. So to interrogate B-regs, which again, as I mentioned, IL-10 is one of the key functions of B-regs, we employed a IL-10 reporter mouse. So these are IL-10 GFP neonates that we then infected with Listeria. And then in order to link this with CD22, we have a blocking antibody against CD22, which I didn't show, but I showed you the CD22 knockout mice, but if you can also use this antibody to block CD22 function as well. And so we either came in with this or an isotype control antibody. So this is flow cytometry data I'm showing you now that shows the percentage of B220 positive IgM and CD5 positive cells. So this is enriching for the B cell subset where predominant amount of B-regs are located, particularly in neonates. And so when we looked at these cells and then looked at IL-10 production, we saw that compared to a no infection control, when you transfer these acetylated non-protective antibodies, there's an upregulation of IL-10 production by B-regs. And I'm going to show that kind of schematically over here to kind of start to wrap this up too, is that when you have these acetylated antibodies that aren't able to bind CD22, these B-regs make a larger amount of IL-10. Then what we did was we came back with our pregnant antibodies, antibodies from pregnant mice, and we showed that, again, before we showed you these can protect the mice, but then also we showed that this now reduces the amount of IL-10 production by B-regs. And then when you block CD22 in these mice, you reverse that. So again, schematically, these deacetylated antibodies then are able to both, they're Listeria specific and then also able to bind to CD22. And because of that, there's an inhibition of these B regs and there's less IL-10 production. We also showed this for IL-10 levels in the serum and the spleen. So it's the IL-10 that already been released as opposed to just this flow assay. And we also did this in a more of a in vitro approach, just using purified B cells and Listeria as well. Then what this really suggested to us was then that B regs and then their IL-10 production are impairing the ability of antibodies to protect against Listeria. So what we found then was that actually when you get rid of B regs, and one of the ways we did this was just using a CD19 knockout mouse that still has follicular B cells, but there's no B regs in this mouse. And we found that now when you transfer these virgin antibodies that have the acetyl group on them, that normally aren't able to protect wild type mice, you're now able to achieve protection in them because you don't have this entire B reg subset that's making IL-10, and these antibodies are now able to protect. And then likewise, instead of just using a CD19 deficient, B reg deficient mouse, we came in with a blocking antibody against IL-10. And we showed here that these virgin antibodies, again, that normally don't protect, are now able to protect when you neutralize the IL-10 receptor. Again, so bypassing the effects of IL-10. So to summarize then, what this means is that when you have high IL-10 from these B regs, you have high bacterial loads and high mortality in neonates. But when you kind of reverse this process by using these deacetylated antibodies, you now have low bacterial loads and low mortality. So then just to summarize the mechanism that we kind of put forth from this paper is that normally there's these acetylated antibodies that are produced during Listeria priming. These antibodies can then be deacetylated during pregnancy by an enzyme called SIAE. I didn't have time to show you this data too, but this requires Fc gamma receptor positive leukocytes that we presume are grabbing onto the antibody and allowing and internalizing them, where SIAE can deacetylate them. Then these deacetylated antibodies are transferred across the placenta and across breast milk, to neonates, where they're able to suppress IL-10 and decrease bacterial burdens and mortality. And of course, this process requires CD22 to suppress B regs. And because of this, we kind of these antibodies are now kind of switched on, that they're now able to protect, versus either passive transfer of these antibodies directly to neonates, or in the situation where there is no SIAE during pregnancy, these antibodies remain acetylated, again, can't bind CD22, and you have unopposed IL-10, high mortality, and these antibodies we've referred to as being kind of like switched off. So from that, we're going to conclude here by just summarizing what I've talked about and then a few future directions, and thankfully there'll be some time for questions. So again, we've kind of pointed to this as being like what we call an acetylation molecular switch, that either the presence or absence of this acetyl group can either allow or prevent binding to these Siglecs. And so because of that, if they have this acetyl group, they're turned off, versus if we can modify them in such a way that the acetyl group is gone, they're now turned on. So we're now my group has multi-pronged approach that we're trying to figure out exactly what's regulating this process and this acetylation molecular switch, and dive deeper into these mechanisms. So of course, one of the questions we have is like, why is this happening in the first place? Honestly, like, how does Listeria infection elicit antibodies that possess this acetylated sialic acid variant? And then again, outside of Listeria infection, broader, is this happening during other immune responses we're very interested in? We're also following up on what cells and receptors are required to deacetylate these antibodies during pregnancy, and is this hormonally regulated, or what are the other signaling intermediators that are required? And then as I'm sure most of you have picked up on now, we kind of have stopped at the point where we know that IL-10 is preventing antibodies from protecting against Listeria, but that doesn't exactly tell us what downstream of IL-10, how these antibodies are protective. So what mechanisms are needed to get the antibody to where Listeria is intracellularly, and what are the antigenic targets that are specifically required for protection, and what are some more of those downstream mechanisms of protection? And again, more broadly, why does this acetylation pathway exist, and what's regulating it? And so, we're currently exploiting, again, why Listeria is a useful model for this type of initial basic science discovery-based research, is that Listeria's life cycle has been super well described, and there's key virulence factors like this LLO that it uses to escape from the vacuole and get into the cytoplasm. that is both functionally very important and also a potential target for antibodies, as well as ACT-A, which I already mentioned, this polymerizing virulence factor, which again could be a target of antibodies. So, and we have from Dan Portnoy's group, who studies Listeria extensively, we have some mutants that again are deficient in these different virulence factors, so we can kind of start to tease apart this biology better. So with that, I'm going to stop. I'd like to acknowledge my lab that's helped with a lot of this work, and it's in the ongoing work. Sing Sing Wei's group, where I did my postdoctoral research, was also paramount to getting this paper completed and published. And Ashley Severance did a lot of the human work. And then we collaborated with Andy Herr here at CCHMC, who's helped us with affinity purification and glycan analysis. And as I mentioned before, the University of Georgia CCRC has been instrumental in doing the glycomics work. Here are my funding sources. And with that, I will stop for any questions. Thanks for your attention.
Thank you, Dr. Erickson. Fantastic seminar. We really appreciate it. Lots of data there, lots of work. So congrats to you and your team on some excellent research there.
You, Rob. Thank you.
Yeah, just fascinating. I want to encourage everybody once again to enter some good questions into the chat box for Dr. Erickson, and I'll go ahead and start with Ashley Rogers' first question here. So Dr. Erickson, Ashley asks, for antibodies from a postpartum donor, are the antibodies acetyl profiles more similar to virgin antibodies or pregnant antibodies?
Yeah, that's an excellent question. So what we have found is that looking at when you look in the immediate postpartum period, which is where we used a lot of the antibodies in our functional studies, the antibodies are deacetylated. And then we followed those mice out to about 7 weeks. So like 35 days was the longest we went postpartum, and we continued to profile their antibodies, and they remain deacetylated. We've done some since the publication, this paper, we've done some follow-up experiments where we follow those mice even longer, and eventually you do start to see this acetylation creep back up. We haven't, though, been able to, in that situation, purify enough antibody to functionally be able to do much with that, but it suggests, though, that these deacetylated antibodies do persist for some time. And also in data I didn't have time to show today, that those antibodies also do protect the postpartum dam if you infect her with Listeria as well. So they're not just protecting the baby, but they're also protecting the mom. So that's
great. Thanks. And I think you also addressed Ashley's second question of, do the antibodies ever revert back? And,
yep.
Great. All right. And my colleague at Sino, Amanda Tamaka, is asking, are NK cells also involved?
It's a good question. We haven't directly tested NK cells, like meaning that we haven't done any like targeted depletion or blocking of NK cells. What we've done is we've blocked or knocked out Fc gamma receptors. So obviously NK cells abundantly express some of the Fc gamma receptors. And in that situation, we have found no effect on the ability of these antibodies to protect. So based on that, I would think that NK cells are not involved. But again, yeah, we haven't directly tested that.
Okay, great. Thanks for that. And Stephen Walimu, forgive me, Stephen, if I mispronounce your last name there, is asking, do you think sialic acid modification can have the same effects on other isotypes like IgM type antibodies?
Yeah, it's a great question. Our group is starting to look at some of that more, like the more early immune response to some of these bacterial antigens. And I don't know if anybody works with IgM here. IgM is actually rather challenging. It's actually kind of challenging to purify IgM. So we have not had a lot of success with being able to isolate the Listeria-specific IgM fraction. But we have a lot more success with getting the IgG and even the IgA, some IgA out. So we have not been able to, I think, successfully answer that question. We do see some interesting kinetics. Like there's actually like a higher peak of this acetylation kind of very early on in the immune response, and then it kind of like tapers off, and then it's kind of held constant. So we're trying to get some idea of like the kinetics and what that means as far as the B cell response as well.
Great. Thanks, Stephen, for that good question. Snow Bajpai, I believe, please forgive me if I mispronounce your name, is asking, are these acetylated antibodies not functional in preventing infection in virgin mothers?
That's correct. Well, it depends by virgin mothers is a bit of a misnomer, right? Because
Yeah.
yeah, so it just depends on the timing of it. So like in virgin mice, like preconceptually in females, yes, the acetylation prevents does not allow protection. And so we've done, I mentioned kind of the postpartum mouse experiment, but we've also transferred these passively transferred antibodies to from virgin mice to just naive adult mice. And again, the ones that came from the virgin mice don't protect in adults versus the ones that came from pregnant mice do protect adults. And so we think there's a similar mechanism at play as far as how adults are protected compared to neonates, but there probably are some important differences. And one of the examples of that is like we focused in on B regs in this study. B regs are actually like really dramatically enriched in neonates as compared to adults. So if you're just kind of profiling by flow, like IL-10 producing with B reg phenotype in a neonate, it's about like on a cell per cell basis, like 20-fold higher than in adults. So there is an enrichment for them. And that was part of the reason we thought they might be playing more of a role in neonates. Versus we think there's probably other immunes, either Siglec and other immune cells that play more of an important role in adults. But those experiments are still ongoing and we're trying to tease that apart.
Great, thank you. Great answer. Appreciate that. Tess Altvader, if I got your name correctly, says amazing work. Can glycan profiles influence the activation or inhibition of neonatal B cells?
Yeah, I guess it depends on the type of B cell. So we have, we focused here as kind of like B regs as a responder cell. It's an interesting question about whether or not these receptors and these glycans are modulating like the follicular B cells or like antibody producing B cells. I think that's a really ripe area for exploration that we've just not had time to explore yet. Because for those that are familiar, like neonates in particular don't mount very good antibody responses. So there's some thought that that's because there's not great T cell help, but there's probably also some intrinsic issues with neonatal B cells. So I definitely think this could be one thing that's regulating them. Is the type of is sialic acid, Siglec, and the sialic acid modifications. So that's a great question.
Great, thank you for that. 7 minutes left, so we'll just keep on plowing through, try to get to as many as possible. So Aaron Wexler is asking, why do IgG undergo FcRn placental transfer, but maternal HSA does not? Maybe you understand that question?
Can you what was the first part of it again?
Why? All right, so I'll just read the whole thing to you. General question in FcRn-mediated IgG placental transfer. FcRn bind both IgG and HSA simultaneously to provide long half-lives. Why then do IgG undergo FcRn placental transfer, but maternal HSA does not? You understand that? Or?
Yeah, I don't have a great and admittedly I'm not a placental FC receptor specialist, so I don't have a great answer for that. I mean, in general, like the process is highly ATP driven and penocytosis of just basically getting a lot of antibodies into these trophoblasts, and then from there you really need the FcRn to bind and stabilize the antibody. Otherwise, if you don't get that binding, the non-bound structures are rapidly degraded. But the FcRn really is stabilizes and protects the IgG so that it can be trafficked across to the basolateral membrane and then secreted and then further trafficked to the neonate. So that's probably not a satisfying answer for their question. But it's an intriguing question.
Yeah, sounds good. Sergio Lopez Briones is asking, delta gamma cells play a role in immune response in intracellular infections. Any idea what happens with these cells in your experimental model?
It's a great question. No, I don't. Yeah, we have. Again, I think another underexplored area in neonates of how the role of delta gamma cells and other more of these like innate like cells are doing. But yeah, we have not specifically targeted those or depleted them. And I don't know, honestly, the literature very well of whether or not those delta gamma cells are expressing some of these sialic acid receptors or not. Like most T cells do not. It's typically more on B cells and other innate immune cells, but those cells are, I lump them in a category of just being kind of weird, but it's something I just don't study very well. But I think it's an excellent question.
It sounds great. And then one last question, we'll go ahead and wrap it up. Garima Ronewal is asking, do you think that women who didn't give birth or women who don't get pregnant, do their immunities actually decrease? You understand that?
Yeah, no, I think that's an excellent question. So there is some epidemiological data to go along with that. So you have to separate it. So actually, so pregnancy itself, while pregnant, is a relative immunodeficient state. And there's multiple reasons for that. A lot of it has to do with mechanisms that are important for maternal-fetal tolerance, so that you don't reject the antigenically formed fetus, like Tregs and other, and actually sialic acids involved in this process too. But and then that's also why one of the reasons we encourage maternal immunization against respiratory viruses is because, like there's a much higher probability of a mom landing in the ICU or on a ventilator, especially late in gestation. So they're like, if they're infected with a virus or get sepsis. So again, there's multiple things that during pregnancy that actually promote infection. However, in the longer term, women that have had pregnant, and I don't know if it's exactly linearly correlated with the number of pregnancies, but women that have gone through pregnancy overall have a more resilience long-term against infections. And so is that because of some mechanism like this? It could be. It's an intriguing question. And it's one of the many things I think is super cool about pregnancy is, number one, you kind of make these more super antibodies, and number two, it kind of sets you up for longer health and success. And women in general are also more protective against infectious disease than men overall, and pregnancy seems to enhance that. So I mean, I think there's a lot of really interesting things to learn about what actually happens during pregnancy. How does estrogen, progesterone affect the B cell response, shape these antibody-glycans? I mean, there's a lot potentially of mechanistically why, but I think the short answer is yes, there's definitely some protective effect from being pregnancy after pregnancy. You got to get through that pregnant window, get immunized if you're pregnant right now, little PSA. You don't want to, we've seen too many moms, one of the reasons we saw a lot of premature babies during COVID. A lot of moms got just very sick with COVID and delivered premature. So this definitely happens a lot.
Great. Thank you for that answer. We had a few more questions. Unfortunately, we've run out of time, so we could put those people in touch with you directly.
Yeah, for sure.
Offline, if that's all right. And so now I just want to wrap things up. I want to thank everybody first and foremost, our attendees. We had a fantastic turnout today, and y'all asked very excellent questions. We do appreciate it. Just to kind of recap where everybody's from, of course, across the USA, specifically Georgia, Indiana, Florida, and Texas, and then in Canada, we had attendees from Vancouver and Montreal. We also had folks from India and Belgium, Jamaica, Pakistan, Thailand, Botswana, Gabon, Spain, and Mexico. So a fantastic turnout. Thank you, attendees. Dr. Erickson, thank you so much. It was a fantastic seminar. I mean, that's a lot of hard work for a lot of data. So congrats to you and your team.
Thank you very much. Appreciate it.
Yeah.
I'm tuning in from all over the world.
Yeah, it's fantastic. And also, I just want to thank my colleague, Priscilla Hernandez. She's the one who actually executed this webinar from a technical perspective. As usual, she aced it. So thank you, Priscilla. And with that, I will say good afternoon, good night, and good evening to everyone. And have a great rest of your day.